before the presentation, let me briefly introduce myself. So hello, everybody. I'm Nobu. Actually, I'm from Hong Kong, and I've been working for the YSI Life Science product for almost 15 years. And besides that, I'm also handling the Xylem analytic product from SI Analytics, ePro, Bellingham, and Stanley. So maybe after this seminar or webinar, if you have some question regarding to the life science analyzer or SI analytic product, ePro or Bellingham and Stanley, you are welcome to send me an email. So let's start. So today the main topic we are going to talk about is the YSI 2900s that used in the laboratory to help our customer to increase the efficiency and productivities. And before that, maybe we have to have some idea about what kind of efficiency or productivity most of the customers are looking forward in their laboratory. So a common understanding is laboratory productivity is also called efficiency and effectiveness of a laboratory at producing results. That means in simple is just count how many laboratory work you can do or accomplish within a given time. However, if we would like to look at this more specific, maybe you have to look at how to maximize your output with minimal time input there. And this is regarded as a kind of process optimization. And from today's presentation, we're going to talk about the YSI analyzer, some of its features and operation principle, how the instrument can be a part to make your laboratory more efficient and more productive, and how the animation enable streaming of the operation, and eventually you can make your staff in a good position, in a good way to fulfill their lab work in daily life. So maybe we should have a look about our instrument we talk about today. This is the YSI Life Science Analyzer. So we have the model 2500 or 29 series. So the difference is just the parameter able for measuring in this instrument. So for this instrument, when you look at this picture, actually we can give some idea how the productivity and efficiency can be raised by working with this instrument. So we will see improved workflow, and then also can put the lab technician a spare time to good use because of the automation, and then the small footprint of the instrument also can make the work efficiency in the laboratory. And also, when you have multiple instruments in a particular region, you can also make your laboratory more specific for specific study or measurement. And also the most important, nowadays we see that no longer customer rely on dropping notes. Those measurement data should be shared among the staff or shared among the supervisor. So we call it traceability. Okay, so let's have a first look about how the workflow can be better managed. Because as I mentioned, the data are no longer just rely on just display on the instrument, the result and the control and setup. You can do it remotely. And there's a different connectivity. Like if you have an online system of the YSI Life Science Analyzer, you can output and incorporate with SCADA or output as a LIMS. And then in order to have a feed control system, you can also have an OPC server option to allow a different kind of expansion of the system and the data how to share among the staff and those people who manage for this process. And then turn a good time for your staff, make good use of the time in the laboratory because the instrument is fully automation. That means you are no longer just to present your single test or single measurement every time in front of the instrument. You can prepare your sample in a, for example, like the 96 sample plate, put them all at once and set the scheduler and set the batch running all these tests by the instrument itself. And then you can do something else. Like, for example, you have another measurement or experiment to do in the lab. You can spare this measurement work and go to the other position. And then we talk about the YSI analyzer. Also, they are not big size, so the footprint is quite small. When we look at the 2900, it is a two chemistry model. So the footprint is about 12-inch wide times 20.5-inch deep and 15.75 height. And 2950, it would just have an expansion model. So it looks as like a computer tower on the laboratory. And as I mentioned, if you keep the same instrument, related one in the same space, so you can minimize the transit time, or the sample can prepare at once for automation measurement, and then you put into the sample tray and let the instrument to run by itself. So it also can erase the efficiency of how, if a customer, they have a lot of experiment they need to do, so they can just put all the batch inside the instrument to run, and then you have a result in short time. And then we talk about sharing data between the staff is also very important. So from the YSI analyzer, you can filter, you can export your data or measurement into different media like the USB drive. You can share your measurement as an Excel file, so for future graphic porting, or even you can export the lib, like I mentioned in the earliest slide. We have different connectivity, so it will make the work more lighter in your laboratory, because you don't need to rely on writing everything on the logbooks. Then you can have a review of this data from a computer or from a network. Okay, then we talk about the YSI, why YSI being called gold standard in the laboratory. So firstly, we have to say that gold standard doesn't mean it is a standard method, but this is just a global recognition or by the end user. So how to achieve this gold standard? Gold standard means this technology have the result are quite accurate and precise, and also it can be reproducible. The most important idea is the measurement is pretty fast. And this is the reason why YSI has been branded as the gold standard. In the past, we have the legacy model 2300 on 2700. So they can have been deployed in different area, different field for different application. So we have a look about this YSI lifetime analyzer. So we will have an idea about how it works, why we call it gold standard. So from this few slide, you will see how the sensor technology and what are the benefits of using this instrument, and a quick review from hardware, and also some possible chemistry combination for this lifetime analyzer. And eventually we go back to the topics, how this 2900 series can enhance your efficiency and productivity for the laboratory. So YSI, they are the first one try to incorporate the oxygen electrode together with the immobilized enzyme. So we have this enzyme electrode. So this is a quite long history. So YSI developed this biosensor in 1975. And we will also see actually YSI has other types of sensor like fluorescent and ion selective electrode. And ion selective electrode also being used on 2950 for ammonium and potassium measurement. This is mostly related to fermentation process. And the principle of the enzyme membrane is actually the membrane. If you are familiar with the dissolved oxygen membrane is actually the dissolved oxygen membrane is with a platinum electrode, and then you have a membrane there, and the oxygen pass through the membrane, and with some stirring, you have some signal, electronic signal detected by the platinum electrode. But for the biosensor, it will be a little bit different because the membrane is with a special 3-layer structures. So we can see with different pore size of this 3-layer structure. The first one will be allowing the analytes, like what we are going to mention is the glucolactase or glutamine, and the oxygen will pass through the first layer. And then the immobilized enzyme. Immobilized means the enzyme being trapped there. It will not lose during the process. And then the enzyme will be react with those of the analyte and oxygen. And eventually there will be hydrogen peroxide complementary with electronic signal produced and detected by the platinum electrode. In a quick formula way, we will see the equation like this. Analyte with oxidation, and the glucooxidase is an enzyme that react with this analyte and producing hydrogen peroxide and the byproduct. And the electrode will sense the hydrogen peroxide and the electron flow. And the more electron flow, that means the higher concentration of the analytes. And then with the algorithm stored inside the analyzer, we can work out the concentration of the analytes. As I mentioned, this is a gold standard. So gold standard 3 conditions must be met. First is a rapid test. And for some specific measurement, customer, they only need very small amount of samples, say from 10 to 50 microliter. And then after the sample being injected into the chamber, you can have a result within 60 seconds. And this is quite accurate. For some of the measurement, we can have accurate with less than 2%, plus or minus 2%, because enzyme and substrate, like a key and lock, only particular enzyme can open the lock. So that's why we have such a high accurate for the measurement. And the most important concept for this is the enzyme is immobilized. It would not be consumed after one measurement. That means the more sample you have, the more cost saving you have per sample. Enzymes are not being used up within the test. And the other advantage of using this analyzer is our measurement principle basically measure the currents. That means the sample, as long as it is made in an aqueous solution or aqueous form, the whatever is a little bit turbid or it is color, it will not be affecting the measurement result with the YSI analyzer. And then we talk about the chemistry capability for this instrument. If we look at the 2500 on 2900, this is two electrode analyzer. 2500 are target for glucose and lactase, and 2900, then you will see we are able to measure glucose, lactate, glutamine, glutamate, xylo, ethanol, methanol, sucrose, choline, galactose, glycerol. So this is a different combination. There's also offer a customer a kind of a flexibility, because not everyone need to measure all these parameters at the same time. And then with the 2950, we extend it to handle the iron selective electrode, ammonium and potassium as well. And our major customer for this analyzer are laboratory in the biotech, bioprocessing lab, biofuels, some food and beverage customer, and clinical market. Okay. So when we look at this slide, we see, wow, this is something different from the picture I saw from the previous slide. Maybe for those of you already has been exposed to YSI biochemistry analyzer, you also know some history about this instrument. The other reason why we are called gold standard is because YSI produced a unit called 23 and then 2300 step plus. They were the first one to commercialize an analyzer for blood glucose analysis. So we have a very good reputation for the diabetic research, and 5 out of 6 of the glucose handheld meter, they would utilize YSI 2300, the FDA version analyzer, as a standard instrument in the laboratory to prove their FDA compliance. Besides then the diabetic research, YSI analyzer are also widely used on food processing, in the pre-processing or during processing or pro-processing as a QC instrument, because it is fast. For example, like some soy sauce customer in Asia, they need to make good use of the glutamine produced in the factory, like those with a higher premium quantity. Maybe they can set as a higher price, so they will send to the retail. And those who need a lower price, maybe they would use something set for catering. And then the best way is to utilize the bioanalyzer to give them a quick result accurate, and then they can decide which bag should be go to which stream of the product life or packaging. And then also for beverage, we have soft drink and juice, beer and wine. And allow me to say for beverage, so for example, like tomatoes, somehow it need to control the lactic acid, because if the lactic acid concentration in the juice are not well controlled, you actually affect the taste of the juice. And then sugar processor, effluent discharge. So this is typically not everywhere will implement this kind of the measurement, but for some sugar manufacturer, maybe some regulation control in the particular country, they need to look at the effluent discharge to see whether there's glucose inside, because glucose is the source of the nutrient, and it could lead to growth of bacteria inside the discharge water. And then this is for medical and clinical research. So our major customer use our analyzer to help them for drug development and do some like a cancer research by studying the cell culture, how to control, to make good use of the some environment or condition to make sure they get a better yield. And for drug development, this is very important because they also need an accurate and fast measurement result. And at the same time, for some drugs, because they are so expensive, they are not allowed to use too much sample. So our analyzer, they could just utilize 10 to 50 microliter. This is a small amount, so it is economic use of the sample for them. And then for physiology, the YSI analyzer also can be used for physiology study, and because some customer maybe utilize this instrument inside the athletic center to check the lactate level of the athletes. For example, in order to control how to create the nutrition list or food list for the athletes. And while the athletes are getting a serious training, they also need to study the lactic acid in the blood to avoid overtrainings. And for certain hospital or certain pharmaceutical research, they also need to check some subjects, the glycerol metabolism, like to check as an indicator of the fat breakdown. Okay, so we go back to the efficiency and productivity topics. So because this analyzer allow automation, that means you don't need to present every single of your sample and wait for it finish and then go for the next one. You can, using the modern vessel like the 96 sample plates, and you can put your batch sequence in any way you want. Just press start. The instrument will run automatically. And of course, in between the instrument calibration, everything also automated as well. And with this instrument, the automation area are mainly located on the station 1. In this picture, you see where the needle-like zipper, the running is the automation position. And of course, there's a manual position. A manual position will be mainly for emergency use. And if we look at the hardware of the analyzer, like I mentioned, it's small footprint, and it has touch screen monitor, and the user interface is straightforward. It's only composed of 6 icons. That means you can configure the instrument and then program with a simple input. Then you can set the instrument ready for use. So it's also good for the lab operator. They don't need a difficult learning curve. They just get a intuitive learning, because the operation and the setting of the instrument is quite easy and user-friendly. This instrument also adapt to different model vessel, so make the automation more easier take place in the laboratory. Okay, so 2900s, we can see they can be set for different combination, and all the result after it's aspired and dispense into the chamber, the result will be displayed within 1 minute. Okay, so this is the user interface. As I mentioned, this is quite straightforward and intuitive. When you start up the instrument, it only shows you 6 icons. So the logic is, first you configure to tell the instrument which chemistry you are going to measure, and then after you configure the setting, and then you just press run, it will start to run by firstly calibrate the instrument, and after it's past calibration, it will start to measure your sample. And then once you finish your sample, of course, you have to check your data. So you click the data icon. And then when you find something, I want to service the instrument, I want to have some other setting to be triggered, and you can just go to the setting as a to have a quick way to change the system. And then for help, from the help page, you can locate some of the video, onboard video, and some document. It is very useful when a laboratory they have a newcomer. It will take a lot of time to read the menu to understand how the instrument works. But for this instrument, if you come across to any difficulty or how to set up the instrument, even like the enzyme membrane installation, by pressing the help, it will have some passage or text or video to show you how you can configure and install your system. And then this is this whole workflow, we have a user management program behind. And for 2900 or 2950, this user management is compliant to 21 CFR Part 11. And here we go, we can see even the system are able to have an auto calibration, but of course you can also override by using a manual adjustment to suit your requirement. For example, you just work on weekdays, then weekend you can stop the auto calibration or make it longer time to save some consumables. Here we go, is the help video. When you click the help icon, you can see there's different types of topics listing down there, like how you calibrating the instrument, how you do the normal changing membranes, if you have an online, how you configure the online, and what daily check you have to do before your works. Okay, so even I mentioned this instrument, we have intuitive user interface. Actually, when you were the first one to configure the instrument, everything inside is quite straightforward. You don't need to remember, okay, which model or which article number I need to set. For example, if you press on the glucose, then the default one, calibrator, 27676 will be displayed. And after you config, the item will be checked when you look at the right picture. So the buffer, when you already select in the first page, the next page, then you see, oh, it is checked already, and you get a cross. So everything is so user-friendly. And then finally, for the data output, you can export your data to USB drive, and using the network or Ethernet cable to access with an FTP to get to the data or the measurement you want. And you can even filter your data, FTP to access the data. And the other good thing is, while the instruments are in operation, you can also have a display to show how the instrument look like. Is it working properly? So while you are doing this, when you tap to the surface, you can know, okay, whether the instrument are doing calibration, if the instrument are in good condition. And for online, you can have an online real-time curve to look like this. So this is the other thing I've mentioned. Besides, then you can connect or export your data through USB or network. You can also use a physical thermal printer to print your data, especially some customer, if they are difficult to have a computer inside the laboratory, or it is far away, and they don't, or even they don't have a network. They don't really need to connect to a network. They just put a serial port thermal printer, then they can print out the result. And the most important thing is, for certain pharmaceutical company, they are not allowed to use USB. So direct printing is the only one they can accept. So the YSI live science analyzer also allow this to connect with a physical printer. Okay, then we go for the application review about the YSI analyzer. This is one of the typical example for online using the YSI 2900. This is a customer in United States. How could they use the online system to control the glucose monitoring, and then eventually how to make sure the perfusion of the CHO cell culture are in good condition. If you are interested on that, I can later, maybe you can email me. I can send you this application notes in details. And then this is the application note available from YSI.com. And we have a special topics for them. Then you can have a look of all this application note. So for this page, you see most of them are related to food and beverage, like the measurement of L-lactate and ethanol in tomato-based products, and then the ethanol determination in beer. Yeah. And if you want to further download this application note, please browse the YSI.com and type YSI 2900 or YSI 2950, and you can link to this page. And also, from some of our application note, you can have some inside idea how the market, commercial market, they control some of the measurement result for the products, like for beer, somehow why they need to monitor the sucrose or glucose. And also, YSI result mostly are compared to the HPLC. And we can have a look from this table and see how closely our measurement are close to the HPLC. But if you are laboratory people, you know, running HPLC, it takes times. When you really need to focus to one or two chemistry, running HPLC actually costs a lot. Besides the time, also the money. But YSI, as I mentioned, is a fast method. So, and also a quick way within 1 minute. So if you consider you have a 50 sample or 100 sample, you just need to have the glucose or lactate reading. Then which one will you use? You will use HPLC or YSI. Most of the customer will select YSI. And then this is another application concern related to bioprocessing series. So it will list out, actually, sample is not so difficult to prepare. As long as it is dissolved in aqueous solution, it can be ready for use in the YSI analyzers. And we also have some condition listed to tell you, okay, from which instrument or from which apparatus you need to use, and then what sample size you have to set in the analyzer in order to give you the optimum result. Okay, then after I talk about so many about the YSI analyzer, some customer, if they never really seen or know about this analyzer, maybe they'll think, oh, this is just most of the instrument, they will have this kind of data export or some flexibility. And I like to show you like this because most of our customer, not only on the commercial side, and they are also on the research. So if you go for Google, the Google Scholar, you just type YSI 2500 or 2900, you will see a lot of paper or research study being listed. That means a customer, they use YSI as one of the instruments to monitor the whole research or measurement. And actually, I have prepared some paper that are free of charge, and later on we will send you by PDF. And after I have expressed so many about the YSI analyzer, and maybe you are still if you still have no idea about this, or you are totally new to this instrument, I will highly advise you to go to YouTube and try to check the link here. Then you will see how's this instrument working, why I said it is really good for the laboratory to raise the productivity and efficiency. And because today we have limited time, therefore there's 4 videos here. Each of them have a link. When you receive the PDF file, you can click on it. However, I have already downloaded one. Then you can have an idea how the instruments actually their workings. Okay, so let's start. It's about 1 minute.
With over 50 years of experience utilizing immobilized enzyme electrode technology, we are proud to introduce the YSI 2900 Biochemistry Analyzer. YSI has an established reputation of being the gold standard for laboratory instrumentation, and our biochemistry analyzers are no exception. Our analyzers are utilized across many industries, including bioprocessing, food and beverage, biofuels, and medical research. The robust enzyme electrode technology allows for little to no sample preparation, as analysis is not largely impacted by color, pH, turbidity, or viscosity. The YSI 2900 Biochemistry Analyzer offers a 96-well sample tray, which accommodates samples ranging from 10 to 50 microliters, full sample automation, manual sampling with syringe configuration, programmable auto calibration events, multiple handling options, and much more.
Sorry.
With analysis in less than 60 seconds, the 2900 Biochemistry Analyzer quickly provides accurate analysis. We believe you will find the 2900 to be the easiest to use biochemistry analyzer available.
Okay, so I think everyone have an idea how's the YSI 25 or 2900 are actually working. So before that, I also need to tell you actually the YSI 2500 and 2900 from the outlook, they are 100% identical. And when you're really operating, you will find the user menu interface are slightly different. And it's 2950 is actually, you can think about it is the expanded 2900 with an additional compartment next to it. And today we talk about so many advantage and how the YSI lifestyle analyzer can be benefits to the commercial or academic user, and how they can be regarded as a good instrument and help the customer to increase their efficiency and productivity. Actually, I have prepared some examples to show you how in Southeast Asia and North Asia, how our end user, they think about why they will select YSI analyzer in the laboratory. So in Hong Kong, actually, we have few customers using, but I would like to point out one customer, which we find it is very loyal to us, like this is from the Hong Kong Polytechnic University, the Advanced Biological and Chemistry Department. They have been using these analyzers for a long time, but of course they are using the legacy model at the very beginning for many years, the 2700, and later on they upgrade to 2900. They keep on using this 2900 to demonstrate and show their undergraduate or for their postgrad research as a lecturing. When they have a fermentation section, they would utilize this to demonstrate to the student, to tell them, okay, fermentation process, what is the nutrient, what's the byproduct, and if you want to increase the yield, what condition you need to control in the fermentation process. And then for Korea, we have different kind of customer, but I would like to pick up one which is quite representative. And this is the one I just recently visited before last week. This is actually from the Daesung Inno Park. This facility, I think they are quite big in Korea. They are mainly for commercial. They have kimchi processing. They have food and beverage, and they have a bioprocessing department. There's a multi-layer industrial area. So they utilize the YSI 25 and 2900 for the amino acid study and research. And because of the expansion in the laboratory, they will soon to get another 2 units from our distributor. That means it will help them to save a lot of time, to help them to make their amino acid with a higher yield. And then LG Chem also have a multiple 2900, 250 for the bioprocessing R&D department. Okay, and then this talk about Malaysia. Malaysia also, we have 2 quite good customer there. The Dexcom Malaysia, Dexcom actually from US. They have been utilized the 4 or 5 units of the 2900 for some kind of study related to diabetic diagnosis tools. And because the instrument also very accurate, so we have IQOQ, and that's why they followed our American and utilize our product for in the research. And CJ Bios, they also have more than 4 units of 2900D for the bio API, active pharmaceutical ingredients in Malaysia. And then Singapore, actually, we have other customers, but I would like to point out the NUS, the National University of Singapore. They also have more than 5 units of 2900 there for running their diabetic and GI research. Indonesia, the PT Chedai is also same as the CJ Bio groups. This is our loyal customer. They have, when they effort to expand their laboratory, they will get the YSI 29 and 2950 series. So the main study is for sucrose, glucose, and lactate monitoring for the process. I think now it's come, we still have some time, so maybe we can go to the Q&A section. Okay, let me have a look. There are quite a lot of questions from here. Okay. Yeah, very interesting, this one. Enzyme wasn't consumed by quantity sample tested, but it will consume by high temperature. Okay, so we have to put it this way. Because when you present your sample to the 96 sample plate, or you present it in the test tube in the manual position, the enzyme is located in between the membrane. When you aspire the sample, of course, if you aspire a very high temperature sample, it could affect the enzyme. But mostly for a fermentation, I don't think it reached so high temperature for fermentation process. So you could relax on this. And the other possibility is maybe for some case if you're concerned the temperature may affect your reading, then maybe you have to wait the sample to reach to ambient temperature before your measurement. Then it will give you a more linear result and a more repeatable result for the samples. Okay. You mentioned 2500 or 2900 is 21 CFR Part 11 compliant. Is this case only for user management or data integrity as well? I think there is something I have to declare that only 2900 is, or 2950, it is 21 CFR Part 11 compliance. That means there is a particular page where you can turn this 21 CFR Part 11 compliant as enabled. And then you will be able to give the administrator rights, calibrator, or set some different level for access for this, or even just to let the instrument as operator use only. That means they cannot change everything inside it. And from time to time, also you can see who's the one to log in to do particular thing like change the calibrator or buffer. So this is talking about related to traceability. And then possible. Okay. Even we set the auto calibration to 120 minutes, the machine keep on calibrating every 3 to 5 minutes. Is it possible to inactive the calibration mode, only make it to do calibration like what we set every 120 minutes? I think this I can reply to you later, because when I need to explain this, it takes a long time, and I have to show some procedure to how you can make this calibration work as you want it. Okay, so move on. Will the sample temperature affect the result? If the same sample are prepared, tested under 25 degrees and 30 degrees, will it affect the result consistency? I think this is also a good question. The instrument, actually, they would calibrate based on the sample size, how many sample, and then the time, the duration, and also temperature dependent. We have to understand enzyme actually work a little bit different and different temperature. So if you talk about your measurement at 25 degrees or 30 degrees and in between, if there's no calibration, there will some difference between your result. So for that case, I think you better, if you all already realize you have such a big temperature change in your laboratory, to me, this is only under what condition it will happen is because, like, if you have 1 or 2 operator in a laboratory, if you have an air condition, it shouldn't go like that unless you have many people gather at the same place. But you talk about 5 degrees, it will be really, it depends how you handle the sample. But if you are aware this 5 degrees different, it's better to do a calibration, because based on the instrument, even 1 degree change, you will have the different result possible. So if you have 1 degree lower or 1 degree higher, actually the auto calibration will be triggered by itself. You don't need to really calibrate. If you already enable the temperature is 1 degree, you trigger auto calibration. Will the environment temperature consume the enzyme? My laboratory temperature is around 25 to 28 degrees. I would say that it is the enzyme is not really consumed by the temperature. The enzyme actually we have a working life. For example, like the glucose, when you install on the YSI instrument, the lifestyle analyzer, actually it can last YSI guarantee 21 days. But for some situation, if you run the test, run the linearity, and you still find the result is acceptable, then maybe you can keep on for 1 month. That's why I said the enzyme membrane is not dependent on the sample size. It is, if you have more sample size within certain period, you get a lower cost. So I think I answered this question already. The zipper clean or rinse between sample to reduce contamination. Yes, actually, when the zipper go to the sample, it aspire the sample, and it is actually will be diluted with the buffer together. And while doing this process, actually there will be a rinsing with the buffer to make sure the baseline are more or less the same as before it start to produce. Before it getting into a mold to measure the sample. So yes, there's some rinsing between the samples. There's can I have more details on ethanol determination in wine? Are there any application on this? Yes, as I mentioned, when you go to ysi.com, you can download all these application notes, and one of it is the application for this determination in wine. Oh, okay. So this last page is, you see there's my email. Some of the question, as I mentioned, is quite technical. I will try to answer you later on. But if you prefer, you can send me precisely the condition you met, and then I'll try my best to answer you. And if you do want to have some marketing information for the YSI License, you can also go to the Xylem Marketing by typing this email info.em@xylem.com. And because this is a recording version, and you will get a PDF file, and I actually prepare Google Scholar PDF file link where you can get some free PDF file to show where the 2900 2950 has been used in different areas. Hope it will be useful for you. I think if there is no further question, then maybe we will end this section today. Thank you for your time.